Cilia are the most important locomotory organ in case of Paramecium and many other protists. Hello friends, welcome to Biology Concern. I am Unimesh and in this video I am going to discuss about locomotion in Paramecium. That is the ciliary movement or ciliary locomotion. So let's jump into the topic. Firstly, the main locomotory organ of Paramecium is cilia, and cilia are classified or subdivided in two broad categories. One is locomotory cilia that helps in locomotion. So locomotory cilia basically helps in locomotion or movement, and other is the oral cilia that help in the filtration of foods. Okay, so basically if we consider the structure of a particular cilia, then we get this kind of structure here. So if we consider this one cilia, so we get this kind of structure. So here is a basal body structure. Then there is a two different structure as this. One is the central fiber, next one is the peripheral microtubule, third one is the matrix, and fourth one is the ciliary shaft. So basically the outer covering or outer part is the ciliary shaft, and inside of this ciliary shaft there is a peripheral microtubule present, and at the innermost layer there is a central fiber present, and all together they combine a structure of cilia. So basically when a cilia resides inside of a cell or plasma membrane, so if we consider this is the plasma membrane, so if we consider this structure is plasma membrane, so we only visualize this portion of structure. So this portion of structure we only get visualized. So this is the outermost portion, okay. So if we observe the total structure, that is the molecular structure, then we found that beneath the plasma membrane, so this is the plasma membrane, so just beneath the plasma membrane there is a presence of air sac. So basically air sacs are present in between one cilia, so there is a two air sac present in between one cilia. So cilia are continuously lined with air sacs, and those air sacs help cilia to hold its structure perpendicular with the surface or with the plasma membrane. So basically the air sacs helps the cilia to form the structure or perpendicular structure, and beneath the air sac there is a network fiber present. This network fiber is known as kinetodesmata, and the basal portion of this particular cilia, that is the kinetosome. So kinetosome is the basal portion of this each cilium, and those each cilium are attached with each other with the help of this kinetodesmata. Okay, so this is the structure of molecular structure of cilia, how cilia is attached with the surface of Paramecium. Okay, so now if we consider the basal body structure, so this is the TS of basal body structure, where this kind of structure is made up of a particular protein known as tubulin protein. So tubulin protein makes this kind of structure, this round shaped structure, and basically it is alpha tubulin. So alpha tubulin composed a structure of a tubule. Okay, so basically these a tubules are complete tubules which consist 13 subunits. So which consists 13 subunits, and those subunits attach with each other and make a particular complete a tubule. So basically there is a structure like that. So these structures are microtubule structure or tubulin structure. So there is a 13 structure are there attached together and makes a complete a tubule. So makes a complete a tubule. So basically here 13 subunits are present in here. But in case when you look about the kind of axonim structure, then we found there is a two different structure. One is a tubule and another one is a B tubule. So B tubule is made by another protein known as beta tubulin. So beta tubulin made up this beta tubule or B tubule, and those B tubules are incomplete tubules. So the structure is incomplete in nature. but the complete A tubule is present here. But in case of the basal body, complete A tubule structure is present, and those are present in triplet nature. So basically 1 2 3 1 2 3 1 2 3, such as that. So basically number one structure is number one microtubule or number one alpha tubulin is attached with the next number 3 alpha tubulin, and so on. So basically there is a complex structure or circular structure present. This structure is known as basal body structure. Whenever we look about the axonim structure, so what is the axonim? So axonim basically composed of a tubulin proteins. So that is the both tubulin, alpha tubulin and beta tubulin. So alpha tubulin makes complete A tubule, and beta tubulin makes incomplete B tubule. So those A and B tubule attach with each other, and those incomplete B tubules are made up of 11 subunits. So A tubule is made up of 13 subunits and B tubule is made of 11 subunits, and those B tubules are attached with the particular A tubule, where each of the A tubule or peripheral A tubule connects with the central A tubule with a structure known as radial spoke, just like a bicycle spoke. And there is another structure known as nexin. So there is another structure known as nexin. So nexin basically present between the incomplete B tubule and complete A tubule. So each of these incomplete B tubule and complete A tubule, the connection is made up of by nexins. So nexins basically hold together all total the subunits, that is the 9 peripheral subunits. So 9 peripheral subunits holds together with a particular protein, that is known as nexin. Okay, nexin. Okay, so basically nexin is attached all the 9 peripheral microtubules or incomplete tubules and complete tubules, and after that there is another structure present known as dynein. So there is another structure known as dynein. So dynein helps the axonim structure strong or flexible. Okay, so the bending of this axonim is restricted by this structure of known as dynein, and dynein are basically two types. There is outer dynein, the dynein present outside is known as outer dynein. Okay, so outer dynein, and the dynein present inside, that is known as inner dynein. So inner dynein. So outer dynein, inner dynein makes a complex that holds the axonim structure still, and it restricts the movement of this particular axonim structure. So basically their movement is still restricted. Okay, and this is the complete ultrastructure of axonim structure, that is the main structure, which we observed the longitudinal section in here, that is the central fiber. So basically those peripheral structures are made up of a structure, and there is a central line or central microtubules is here. So this structure is known as 9+2 array or 9+2 arrangement. So basically these structures makes a complete structure, and this structure is known as axonim structure. So whenever axonim is attached with this particular basal body structure, they make a cilium structure or cilia structure. So now the part of the movement. So basically movement of Paramecium, the movement of Paramecium are two types. First one is the creeping. So creeping is the movement where Paramecium attached to a substratum, that is the substratum. Any kind of solid surface that is present inside of water is known as substratum. So basically when this Paramecium is attached with this substratum, they likely crawl and with this cilia. Okay, so basically they crawl with cilia, and now they are moving forward. So from here to here they are moving forward. So this kind of structure or this kind of locomotion is known as creeping. In swimming, there is a metachronous movement observed in the ciliums or cilias. So there is a numerous cilia present outside of the particular paramecium, and those cilia are move with all together, and this kind of movements is known as metachronous movement. So basically whenever they basically cilia, so if we consider this is a cilia structure, so cilia beats all together, then it creates a force on water, and according to Newton's third law, the water it reacts opposite way, so the paramecium gets moved or swim. Okay, so basically this forward stroke is known as effective stroke, and whenever this forward stroke is done, then there is a accumulation of energy needs to be done, because without the accumulation of energy there is no second beating or second time beating. So the cilium are now retracted to its original position, and this position is known as recovery stroke. So basically they recover the strength, they recover the energy, that is the static energy. So whenever ciliums push forward or strike forward, they create a force on the water, and the water creates the opposite force towards the paramecium, and the paramecium moves to the opposite way. So this kind of movements is known as swimming. So this is all about ultrastructure of cilium and the movement, that is the creeping and swimming. So in creeping, cilia are basically crawl itself, and paramecium like just crawl on a substratum, but in case of swimming, they produce an effective stroke and recovery stroke, by which they create a force on water, and water creates the opposite force on paramecium. That's why paramecium get moved. So this is the locomotion of paramecium. I hope you like that video. If you like that video, please hit the like button, don't forget to subscribe, and thanks for watching.